Hi, Dr. Coertzen. Sorry, we're having a little bit of technical issues this morning.
Problem. Okay.
I hope you're doing well.
I'm very well, thank you.
That's good.
Can I share my screen so long?
Yes, please do. Okay, perfect. So while Dr. Coertzen is just busy sharing her screen, I'd just like to wish everybody a happy International Women's Day. And I would like to apologize for the technical issues that we had this morning. I'm standing in for Shruti as she's not in today. And I'd like to introduce Dr. Dina Coertzen to you. She's a part-time lecturer and a senior research scientist at UP, at the University of Pretoria in South Africa. So on to you, Dr. Coertzen. So she'll be sharing about the oxidative stress of the malaria parasite.
I'm just not sure if you are able to see my screen.
We are now, but put it in presenter mode. Let's see if it disappears.
And
perfect.
Is it fine now? Okay, just checking if you can see my laser pointer.
Yes, we can see your laser pointer.
Okay, so thank you. First, I'd like to thank Shruti and the team for inviting me today and giving me this opportunity to present some of my research. So as I was reading the World Malaria Report beginning of this year, I realized that the impact that COVID has had on malaria elimination strategies over the past 2 years due to COVID-related disruptions has been quite significant. So it's now more apparent than ever that there has been a significant decline in eradication of this disease, particularly as over the past 2 years, not only due to COVID, there has been a significant increase in the amount of deaths and numbers of cases that we've seen with this disease. Now, COVID wasn't the only impact that influenced elimination strategies, as several reports have now been launched in which independently formed K13 mutants, meaning carrying resistance towards currently used artemisinin-based combination therapies, or ACTs, have now been found within the African continent. And therefore, there is now more than ever a need to drive new antimalarial therapeutic strategy delineation and drug discovery for treating this disease for elimination in the long run. The caveat with current antimalarial therapies are that ACTs only are effective in eliminating the asexual blood stages responsible for causing disease symptoms. Now, these compounds are known as target candidate profile 1 compounds, but due to the progress that's been aimed towards eliminating the disease, the field has taken a shift in developing compounds that potentially target the gametoside forms of the parasite. So these are these intraerythrocytic parasites that are responsible for disease transmission. And compounds that then target these gametoside stages are what we refer to as TCP5 compounds. So this will not only eliminate onwards disease transmission, but also help prevent the spread of drug-resistant phenotypes. Now, in a review that was published in our lab last year, the chemical characteristics that are defined for compounds that have both TCP1 and TCP5 characteristics, or TCP5 only, is that they should have a distinct biological activity profile, in that they either target stages of the parasites differentially, or that they target different biological processes, or inherent that these compounds then have some polypharmacological activity within the different stages of the parasites. And these are then the ideal kinds of profiles that you want to then have these compounds to carry forward development into new antimalarial combination strategies. So With identifying compounds that are active against the gametophytes, an immense effort has been employed over the past 15 years. So large compound libraries have been screened, but in generally, we know that compounds tend to have decreased activity against these non-proliferating gametophyte forms. In a paper that was published in our lab last year, we can see that generally known or clinically relevant antimalarial drugs are not generally as active against gametophyte forms as compared to the asexual stages. However, after these immense screening projects, there are several compounds that have now been entered into clinical studies that have potential transmission reduction activity. And these are highlighted on the MMV website. So these are not only novel chemical scaffolds, they actually have novel chemical mechanisms of action as well. For example, that they target PF-ATP4. One compound inhibits acetyl-CoA synthetase, and there's the elongation factor 2 inhibitor, and even some internal secretion pathways. But we know from our past experience in developing antimalarials that the clinical success of these compounds in terms of transmission blocking only is not guaranteed. And we have to have a continuous pipeline of novel drug development. Now, one of the caveats or one of the challenges we have in actually targeting gametophytes is that we are limited by our biological knowledge of these gametophyte parasite forms. However, we do have some limited indications in terms of what's happening on a genomic, metabolic, and proteomic level. And we know from experience that these parasites have some clear, distinctive biological processes that are being either upregulated in the asexual stages and the gametophytes. For example, that of hemoglobin metabolism. We know the asexual blood stages have a highly upregulated hemoglobin metabolism, as it's using this molecule as a nutrient source to aid in its rapid growth and proliferation. Whereas in the gametophyte stages, this metabolism is subsequently decreased as the gametophyte forms mature, with the stage 5 forms of the gametophytes basically having no hemoglobin metabolism. Now, this ties in with the energy production metabolism of the parasite, and we know that the asexual blood stages favor a more anaerobic fermentative ATP production pathway, in that it upregulates its pentose phosphate pathway and glycolysis. And interestingly, the gametophyte stages are doing a complete shift, favoring a more aerobic, respiratory favored ATP production pathway by upregulating the mitochondrial TCA and electron transport chain. Now, this is all interesting in terms of distinctive biology, and coincidentally, all these pathways tie into one distinctive regulatory pathway that is quite essential, particularly in the asexual blood stages, and that is that of redox metabolism. So we know that as a result of hemoglobin metabolism, the asexual parasites are quite oxidatively stressed, which makes sense why the enzymes involved in maintaining redox metabolism are so upregulated within these stages. But what is interesting is that it seems that these enzymes is just as important within the gametophyte stages. And this piqued our interest in terms of a drug discovery perspective in potentially exploiting redox metabolism for drug discovery purposes. Now, just a little bit of background on oxidative stress in the parasites. The asexual parasites do have an increased amount of inherent oxidative stress production, mainly as a result of hemoglobin digestion, but there's also some cytoplasmic and mitochondrial respiration-based contributions that contribute to ROS. And there's a fine balance that has to be maintained within the parasites, in that an overabundance of ROS can lead to cell death by directly damaging cellular structures such as the lipids, DNA, and even proteins, alkylating them for degradation. But ROS is also an essential molecule for actually mediating essential metabolic processes within the cells by mediating S-glutathione and S-nitrosylation reactions. And these are actually important in mediating metabolic flux within these parasites. So as a result of the fact that redox metabolism ties in so well with key essential pathways within the parasite, the nature of this metabolic pathway is extremely complex and intricate. But for this presentation, I've simplified it very much as seen in this diagram here. Quintessentially, if we have ROS production in the cells in the forms of superoxide anions, these are reduced into hydrogen peroxide by the first line defense in redox homeostasis regulation in the parasite, which is referred to as our superoxide dismutases. The hydrogen peroxide is then further reduced into water and oxygen by two thiol-dependent reducing systems, the most abundant and predominant one being that of glutathione. So glutathione is reduced it is oxidized from its reduced form by glutathionase transferases, and then this can be re-reduced into reduced glutathione using NADPH as the main reducing source. We also have peroxyredoxins involved in this reaction that mediates the oxidation of a reduced form of thyrodoxin into its oxidized form, which is then also re-reduced by thyrodoxin reductases. And then we also have, importantly, various other proteins that can mediate oxidation-reduced reactions via their surface cysteines, such as our peroxyredoxins and our glutaredoxins. Now, interestingly, our red blood cells also have a very similar base redox reaction or redox homeostasis reaction, utilizing catalases instead of the glutathionase transferases that we see within the parasite. But generally, our GSH is still our main reducing agent, is still our main thiol reducing agent, with NADPH being our main electron-based reducing agent in the cell. Because of the parasite's dependence then on glutathione and NADPH, we know that pathways generating these metabolites are also quite essential in the asexual stages of the parasites, meaning that we have NADPH being produced by the pentose phosphate pathway, as well as through the mitochondria, and glycolysis, which is tightly interlinked with that of glutathione metabolism to generate glutathione. But as I'm sure everybody in the audience knows, that we are not dealing with some normal parasite. Yeah? This is a very clever parasite in that it has developed ways to quintessentially hijack the red blood cells reducing power as well. So it can import reduced glutathione as well as reduced NADPH to supplement its oxidative stress pathways. And with that, it can export the oxidized form of glutathione as well as excess hydrogen peroxide. Now, what does this mean in terms of the gametocyte stages? What we know is that the asexual stage redox metabolism is well defined, and we know how it exploits erythrocyte in terms of aiding its redox oxidative stress maintenance. But if you take a look at gametocytes, as highlighted previously, gametocytes, particularly the mature stages, are devoid of hemoglobin digestion, and they do not have, or it's presumed that they do not have, an actively respiring erythrocyte that can aid in redox detoxification. So we were faced with the question, why do we have these upregulation or maintenance of redox metabolism enzymes in the gametocyte forms if these parasites or stages of the parasites do not have direct redox or RUS production in terms and oxidative stress? So if we take a look at this cascade in terms of the proteins involved in redox regulation, and we pull apart all the components involved in redox homeostasis in the parasites, we can divide these into several classes. So these include our superoxide dismutases, which are equipotently expressed during the asexual and the gametocyte stages, our peroxyredoxins, which are quite downregulated during the gametocyte forms, our glutaredoxins, which have low-level increases in gametocytes, and then our glutathione regeneration enzymes. So these are enzymes involved in glutathione production, as well as the enzymes involved in glutathione oxidation and re-reduction in terms of glutathionase transferase and reductases, which are expressed at low levels across the stages. The thyrodoxins, on the other hand, seem to be as important in the asexual stages as they are in the gametocyte stages, and several transcripts have shown to be highly or abundantly expressed during these gametocyte forms. Now, we are quite limited in what we know in terms of biologically what these enzymes are doing and their essentiality, not only in the asexual stages, but nevertheless in the gametocytes. However, some limited essentiality studies have been performed in the past. And what was interesting is that most of these enzymes, despite the importance of redox metabolism in the parasites, have shown that, for example, the superoxide dismutases, the peroxyredoxins, and the glutaredoxins are quite important in the asexuals in terms of maintaining parasite growth and proliferation through mediating oxidative stress. But what was interesting was the enzyme responsible for glutathione reduction, or glutathione reductase, was dispensable in the asexual stages. But intriguingly, it was cultures in which this gene was attenuated in gametes did not have gametocyte maturation. Similarly, with thyrodoxins, one particular thyrodoxin enzyme, our thyrodoxin peroxidase, we could see that the gene was dispensable in the asexual stages, and again, did result in attenuated parasite growth and maturation when tested on gametocyte forms. But these experiments are limited, and it gives us little insight into terms of what are the roles and the essentialities of these enzymes in total when comparing redox homeostasis between asexual and gametocyte forms. But the fact that some of these enzymes were deemed attenuated, not only in the asexual stages, but also in the gametocytes, further intrigued our interest in potentially exploiting the parasite's redox biology as a dual-stage drug target. So when we looked into potentially targeting these pathways, we first looked into what compounds have been investigated that could be exploited or that could be used to exploit oxidative stress or redox homeostasis within the parasites. And the multiple compounds have in the past actually been generated or been shown to be redox active compounds. And we can divide these into more or less three classes. The first class are compounds that basically induce oxidative stress by indirectly targeting a particular pathway that leads to accumulation of reactive oxygen species. For example, our aminoquinoline compounds that inhibit hemoglobin metabolism and hemosome formation are shown to be such oxidative stress-inducing compounds. So when we looked at activities of these compounds across the asexual stages as well as the gametocyte stages, we could see there were some compounds that showed good potency against gametocyte stages, but overall, the aminoquinolines are known to be inactive against the gametocyte stages. A second class of redox active drugs are then compounds that directly induce oxidative stress, and these are referred to as oxidant drugs. The most well-known example of this are the artemisinin, because these compounds have shown to be directly able to form reactive oxygen species in the form of hydrogen peroxide and superoxide anions, while it's also displaying polypharmacology in that they are shown to not only be involved in inducing DNA damage, but also directly alkylate certain proteins, linking them for degradation. Now, the artemisinins do have some activity against the gametocytes, but the fact that we routinely observe this loss in stage 5 activity is confirmatory of their dependence on free hemozoin for their activation, meaning we have cleavage of this N-peroxide bridge that results in the release of the superoxide anion. The third class of oxidant drugs are then the redox cycler drugs. So these are compounds that can alternate between an oxidized and a reduced form. The most well-known example of this is then methylene blue. So methylene blue is a compound that inhibits glutathione reductase, thereby depleting intracellular NADPH and also simultaneously producing reactive oxygen species while it's cycling between its reduced and oxidized forms. So these compounds have some good activity against all stages of the parasites, including asexual all the way to mature stages. And we were particularly interested in that of methylene blue. and it is routinely shown on various gametoside assay platforms to have potent activity against mature stages of the parasites. So because of the interest that we had in redox homeostasis and the expertise in our lab that we have in terms of screening for compounds as well as metabolic or gametoside activity, we got involved in a project that was hosted by Professor Richard K. Haynes. So this was a SAMRC flagship project with the aim of developing drug combinations comprising of oxidant and redox drugs for the treatment of malaria, TB, and related diseases. And as a postdoc on this work at the time, my role was specifically then involved in screening the activities of a large range and a wide variety of redox active compounds against both the asexual and the gametoside stages. And we screened various compounds with known chemical structures as well as novel chemical structures, as shown in some of the publications I've highlighted here. But I'd like to focus on a particular set of these, which include the 10 amino DHA piperazine derivatives of artemisinin. So as shown on previous slides, so artemisinins did lack activity against mature stage 5 gametoside. But these 10 amino artemisinin derivatives, particularly artemiside and artemisone, did show improved activity against gametoside-to-cytosol forms and were therefore prioritized for further medchem studies. From these, we derivatized a DHA piperazine, phenylurea, a sulfonamide, and a rilamine, all of these showing good activity, but the DHA piperazine molecule itself was then prioritized for further downstream medchem optimization. And excitedly, we observed that these compounds, these DHA piperazine derivatives, showed significantly improved activity, not only on the asexual level, but also on the gametoside level. So the chemists got very excited in terms of the activities of these compounds. But as a biologist, we got excited and started asking about what was this compound doing that enhanced its activity, not only on the asexual stages, but particularly then on the gametoside stages. So due to the increased activity we have with these compounds, we also investigated their activities in terms of cross-resistance towards K13 field isolates. We also showed their cytotoxicity. We also showed that they decreased cytotoxicity and improved activity against porozoites. And the added advantage was that these compounds had no hydrolysis to DHA, which has shown some in vivo cytotoxicity levels, and these compounds had improved solubility and stability, which has been something that plagued artemisinins in the past. And this work was published in 2020 and was performed in collaboration with Elizabeth Winslow's lab and that of Dennis Carr's lab. So, like I mentioned, from a biology perspective, we were interested to see what were these compounds doing in terms of their redox activity in the parasites. And as originally stated, the aim of the project was to use these compounds in a combination-based assay platform. And then what we further wanted to do was to delineate what was the potential mechanism of action of using these compounds, these oxidant and redox active compounds in combination. So we could show that if we combine these compounds in a 1-to-1 ratio, we did see a decrease in the in vitro IC50 values against both the asexual blood stages and the gametoside forms. But when we did full isobol analysis, we could show that these two compounds in combination in a 1-to-1 rate or in a full isobol analysis showed an additive effect against the asexual blood stages. But with the gametoside stages, intriguingly, we saw that we had a clear synergistic interaction. So this indicated to us that the compounds, if they would, this combination of an oxidant and a redox active compound was indeed perhaps exploiting some of the required redox metabolism parameters required by gametosides. And it confirmed to us that this redox metabolism was indeed a good drug target for exploiting in these gametoside stages. So at this stage, we needed to look into investigating what was the mechanism of action of these two drugs used in combination. And we looked into the wide variety of available assay platforms that are currently used to investigate oxidative stress within the parasites. So firstly, this work is pioneered by the lab mainly of Katja Becker at Justus Liebig University in Germany. And they can quantify oxidative stress in parasites on both a file level, on a ROS level, and then also they developed a redox sensor in order to quantify and indicate ROS in these parasites. With targeted file measurement, they used good old DNTB, which I'm sure we all remember from our third year practicals. This molecule can then indicate the total level of reduced glutathione, as well as mediate between oxidized and reduced glutathione forms. And it's also used as a means to indicate the total file content of a parasite lysate. File tracker violet is also a fluorescent dye that is used that indicates an increased fluorescent signal in the presence of, that indicates an increased fluorescent signal in the presence of a strong reducing condition within, a strong oxidizing condition within the parasite. For this, I showed diamide, which is an oxidizing agent that basically reduces all files within the parasite and therefore gives an enhanced fluorescent signal if we have increased oxidative stress levels within parasites. And recently, a technique was developed by Darren Creek's lab in Australia, in which they could quantify the amount of reduced versus oxidized glutathione within the parasites using LCMS. And this is really a breakthrough in the field, as the exact quantification of these two file levels has always been limited by other assay platforms. In terms of ROS detection, the most routinely used dye is that of DCF-HDA. And this dye is a good dye that can be used on both a plate reader format, it can be used microscopically through confocal laser microscopy, and then also in flow cytometry analysis. But all these assay platforms, in terms of determining file content as well as ROS detection, do have their limitations and are mediated or are responsible for forming some form of artifacts within the parasites. And therefore, the lab of Professor Katja Becker, actually developed a transgenic parasite line in which they could quantify the amount of reactive oxygen species within a parasite by incorporating a human glutathione reductase as a reporter molecule that was linked to a superfluorescent green GFP protein. So what they could show was that you have a ratiometric increase in the amount of signal you have in this parasite line in under severe oxidative stress conditions, as shown here with diamide, which increases the ratio significantly as compared to a fully reduced sample as shown with DDT. Now, this cell line has been used to validate that there is some form of oxidative stress that is induced with some various oxidant drugs, such as DHA and the triazoline molecules. So, in investigating what the two compounds, artemisinin and methylene, were doing in combination with each other, we visited the lab of Prof. Katja Becker in Germany, and we tested our compounds on the line. Unfortunately, methylene blue, being a highly reactive molecule, directly interfered with the GFP reporter protein in its recombinant form, and therefore we could not test this protein in combination on the lines. But when we tested our lead compound artemisinin on this line, we again observed no significant increase in the amount of reactive oxygen species or oxidative stress being induced in the parasites over a period of 24 hours. Now, unfortunately, this did not indicate to us exactly what these, unfortunately, because we couldn't use this line to indicate what was happening on a parasite level in terms of oxidative stress reduction, and we were limited to only testing this on an asexual line, we had to interrogate some other assay platforms as well. So we came back to South Africa and we started off by optimizing an assay platform using parasite enrichment and the good old fluorescent ROS indicating fluorescent dye DCF-HDA. And we could see that in a similar time-dependent and concentration-dependent manner, we did see an increase in the amount of ROS that was produced on both an asexual level and a gametocyte level. And we could show that there was a significant increase in the amount of ROS produced when the two compounds are used in combination with each other on both parasite stages as compared to the compounds being used as monotherapies. So this did indicate to us that artemisinin in combination with methylene blue did result in some increase in intracellular ROS production. But any biochemist would tell you that DCF-HDA is not a solid indicator and quantifiable molecule in terms of measuring intracellular ROS, as this molecule is known to form artifacts with various of the reactive oxygen species, but also in a cell culture system where we have so many heme molecules available, DCFDA does have some off-target interactions there. So we had to cross-validate these results with an alternative assay platform. And given the success that our lab has had in the past using a luciferase-based assay platform that was able to give a very strong signal in the context of low parasitemias and gametocytemias, we used the Promega ROS Glow hydrogen peroxide luciferase-based assay platform. And here we could corroborate what we saw on the asexual stages, in that we had a significant increase in the amount of reactive oxygen species in the form of hydrogen peroxide being produced on the asexual stages, but then also a lower level increase observed on the gametocyte forms. We also wanted to confirm what was happening in terms of file production. And again, we used a luciferase-based assay platform that could quantify a shift in reduced versus oxidized glutathione. And this assay also confirmed that we had a decrease in the amount of reduced glutathione in both the asexual and the gametocyte forms. And we could corroborate this using microscopy assays or microscopy analysis with the fluorescent dye file tracker, where we saw a decrease in the amount of signal we had with these compounds used either as monotherapies as well as in combination, which again showed to us that the amount of ROS and reactive oxygen species that was being produced in the parasites did result in a shift in terms of reduced to a more oxidized status in terms of files within the parasites. But again, we were still asking that questions we had from the beginning. Why were we having this additive interaction on the asexual stages versus the synergistic interaction we had on the gametocyte stages? What biologically was happening in gametocytes that wasn't happening in the asexual stages that made redox homeostasis so much more sensitive towards interrogation of these two drugs in combination? And this then brings me back to some of the key points that I highlighted in the beginning of the talk, where we have this big shift from an anaerobic fermentative metabolism in the asexual stages to the gametocytes being more dependent on mitochondrial-based metabolism for energy production. And we know the electron transport chain are also responsible for inducing some form of reactive oxygen species. And the question that we asked was, could the fact that we have the increased metabolism within our mitochondria within these gametocyte stages be responsible for the increased susceptibility to the gametocytes when they are interrogated with a strong oxidizing environment, such as the combination of artemisinin and methylene blue together? So we had to interrogate what was happening on a mitochondrial level in terms of oxidative stress on the parasites, particularly the gametocytes. And we used two mitochondrial indicative dyes. The first dye being mitotracker CMX ROS, which has a decreased signal in inducing in a mitochondrially oxidizing environment. And we could quantify this not only microscopically, but also through flow cytometry analysis, where we saw a significant decrease in mitochondrial signal when the two drugs were used in combination as compared to an untreated sample. We also used the mitosox dye, which is a dye that fluoresces, that indicates a stronger fluorescent signal in the presence of a lot more reactive oxygen species in the mitochondria. And we could see again that with the two drugs in combination, we observed a significantly stronger signal in the mitochondria in terms of the two drugs alone. And this is kind of leading us to this kind of led us on to answer our question about what was happening on a metabolic level in the gametocytes in terms of mediating their oxidative stress. So to end of the presentation, I just would like to summarize some of the key work and highlights of this the work that I've done so far. We have been able to use two compounds, artemisone and artemiside, in terms of interrogating parasite redox homeostasis. So using these two compounds as a proxy for inducing oxidative stress, we would like to then further interrogate what is happening on a cellular level in terms of oxidative stress production and in terms of flour measurement, and then also confirm our preliminary data in terms of what is happening on a mitochondrial oxidative level, particularly on the gametocytes. So this brings me to the question that I posed in the title of my talk, in that we can use these platforms or these assays as a mean for indicating, as a tool for indicating not only parasite viability in terms of their the viability in terms of the oxidative status, but we can also then use these assays as a guide to drive drug discovery, in that we can validate the mechanism of action of novel chemical matter. And we can also use these platforms or assays as a means to do biological investigations on the parasites, particularly on the poorly investigated gametocyte forms. So in future, we hope to further characterize the redox homeostasis profile of gametocytes as these stages are poorly investigated. And we aim to do this on a genomic, a proteomic, and a metabolic level to interrogate what are the roles and the essentiality of the genes of these the genes and the proteins involved in redox homeostasis in these parasites, but also to interrogate what is happening with the amount of metabolites directly involved in this pathway, but also interlinked pathways such as the pentose phosphate pathway, glycolysis metabolism, and even glutathione metabolism. We also would like to quantify and confirm what are the reducing power sources that we have within the asexual stages as compared to the mature gametocytes, in the sense of confirming are these parasites more dependent on glutathione or thyrodoxine as the main reducing agents, and then compartmentalize the source of NADPH production in both these stages as well. So lastly, I would just like to thank the team and the great scientists that I've been working with in order to get this work off the ground, particularly the M2PL lab that is run by Professor Lemery Burkholz. She has been my supervisor for a long time, and I have learned immensely a lot in this lab, and received all the training and exposure that I've had as a result of doing my postdoc and my PhD in this lab. And along with that, the great team that has been working with me in this lab, particularly Janet Rieder and Mariette van der Vat, and then also all the technicians and students that have worked on this project in the past. I'd like to say a particular thank you to Professor Richard K. Haynes, which has been a close collaborator on this work and also provided lots of insight and guidance, driving the questions and the answers that we've gotten, as well as Professor Katja Becker and PhD student Katrina Shu for allowing me to visit their lab. So that is it from my side today.